So now we're going to look at primary and secondary responses based off the immune system, right? So specifically when we want to look at it, we want to look at our adaptive, and that branch involving our antibodies as what? It's the humoral response, right? So we want to look at the humoral response on these primary and secondary response, all right? And so there's a very classic experiment that's been done where they compared the responses, and by immune response, I'm referring to the generation of antibodies, based off of exposure to two different strains. So we're going to label strain A here in green, and then we're going to label the second strain B in pink, right? And there's going to be different exposure times, right? So we're going to have two exposure times. At 0, we're going to expose only one strain, and then at day 28, we're going to compare two strains at the same time and actually measure the responses, right? So this is a classic experiment done in order to demonstrate their body's memory that's created after exposure of a specific pathogen, right? So if we look at strain A, at day 0, so we can say day 0, you have 1 exposure, so this is time 1. And then at day 28, we have our second exposure. All right. So you have two different exposures that's generated. So at 1, we're going to only have one pathogen. So only A is going to be present. And then at 2, we're going to have A and we're going to have B, right? And this is, again, used when you use these different strains, it's used, some of them are used just for controls, right? So on day 0 from this graph, your body, when it's exposed to this strain, is at war right now, right? So your adaptive immune system, specifically your cell-mediated immunity, becomes active and it wants to destroy everything, right? And after a certain period of time, you have a production of antibodies, right? And so this graph here, so the Y is showing you the concentration of antibodies in your plasma, and the X is showing you the time in terms of days, right? So it takes a couple of days in order for an actual generation of an immune response here. And that's why we see like a sort of an increase and then back down to a decrease here, right? And so we generated antibodies, and now the cells against that specific strain A has created memory, right? The way to prove this is where they used a second experiment here where they reintroduce that same strain, right? So that original strain that has the memory now, right? So we have memory here. And then they reintroduce a completely different strain that is just as infectious, but just a completely different cell type that should generate the same level of immune response as sort of as a control here. So what happens here is when the strain B is exposed here, it generates at day 28, you see a little bit of a lag again, and you see an increase, and it goes back down, right? Almost identical in terms of response with the strain A. However, strain A is different now. So when strain A now gets introduced, the response is not only faster, but the production of antibodies is much greater. And so this graph is basically showing you how after exposure of that primary strain, the reason why we can never, for example, get sick from the same pathogen, same strain of pathogen, is because of this adaptive response from the humoral immunity. So first exposure, you generate these antibodies, right? Now the cell has a sort of memory. This memory can be now determined based off of this experiment. Because now if I reintroduce that same strain back in, you see a significantly increased level of antibody production and at such a rapid response. There's not a delay anymore, as we noticed at the first site, right? So the first site probably took about 7 days, give or take, in order to have this response. Here, on the other hand, you see after day 2, probably maybe day 1, you have this significant increase in antibody production, right? And so this is a great example of showing you how our bodies is capable to adapt and prevent any kind of infection that is being created based off of this experiment, this classic experiment that was done, I don't know, maybe 50 years ago.